and its uses in today's technology, presented by Amanda, Kimberly, and Vanessa.
GFP was originally discovered by Osamu Shimomura. He spent many summers at the Friday Harbor Laboratories of the University of Washington, collecting the Equior Victoria jellyfish for research purposes. He was fascinated by the glow his jellyfish emitted, and decided to determine what was responsible for the glow. In the early 1960s, Shimomura continued his research at Princeton University and successfully isolated the two proteins responsible for the glow from his Equior Victoria jellyfish: Equiorin protein and GFP.
Equorn, composed of multiple alpha helices, is known for emitting a blue light when calcium ions are present. These calcium ions function as cofactors.
GFP, also known as the green fluorescent protein, is most notably known for its beta sheet barrel structure and its ability to glow when exposed to UV light. Proteins that emit a glow when exposed to light of a different wavelength are considered bioluminescent. When the GFP molecule is exposed to the blue light emitted by the photoprotein equiorin, the photons from the blue light are absorbed by GFP, and GFP emits a photon which corresponds to a wavelength in the green visible light spectrum. The green light that is observed is due to the change in energy that is produced from electrons jumping between orbitals.
The green light that is observed is due to the change in energy that is produced from electrons jumping between orbitals. By looking at excitation and emission peaks, it can be seen that GFP absorbs photons of a wavelength of 395 nanometers and 475 nanometers, while emitting a photon of wavelength 509 nanometers. A shift between potential energy surfaces result in the emission of a wavelength lower in energy than that of the photon that excited the GFP molecule.
To understand how these photoproteins fluoresce, let's look at their components. In general, compounds that fluoresce are collectively called fluorophores. The wavelength of the light emitted, and therefore the color emitted, depends upon the chromophore, the functional group embedded inside the fluorophore. The specific colors observed depend on the type of atom or atoms attached to the chromophore complex, called oxochromes. GFP only needs to be exposed to blue light to fluoresce, unlike aquarin, which would only glow in the presence of calcium ions. This made GFP more useful than aquarin, and was a characteristic that Martin Chaffee exploited.
Martin Chaffee learned about GFP and its amazing properties, and it gave him an idea. Why not use GFP as a marker to locate certain proteins and observe structures?
Martin Chaffee inserted the gene that encoded for GFP into the promoter of Senior-Deptus elegans plasmid. He was successful in incorporating his plasmid into the organism, producing GFP and thereby fluorescence. But what if you want to observe multiple components at once?
As mentioned earlier, the oxophore part of the chromophore is responsible for the type of color emitted by the fluorophore. That's where Roger Chen comes in. He realized that an alteration of the oxophore could change the type of color emitted by the fluorophore. An alteration of the chromophore can result from introducing gene mutations into the GFP DNA sequence. As you know, GFP glows green when exposed to UV light. The reason for the change of the light emitted is due to the change in energy resulting from the movement of an electron to a different energy level. This energy difference is fixed for every chromophore's oxyphore. In order to produce different colors of fluorescence, a structural change in the oxyphore can produce a different energy transition. This change in molecular structure results in a different wavelength of light emitted compared to the wild-type GFP. Therefore, GFP can be modified to emit a broad spectrum of colors which can be simultaneously viewed under UV light.
Researchers can study cellular compartments and determine whether proteins are present using GFP. For example, one use of GFP is to use it as a tag for locating and determining gene expression. In a simplified case, a gene can be modified to contain the GFP DNA sequence, and if the gene has expressed properly, the produced protein will be observable under UV light. By simply exposing the system to UV light is adequate enough to determine whether or not a gene has expressed properly and to locate where the protein is in a system. Noted applications of GFP that impact today's life can be seen in today's research. This includes the following: research in cancer, neurobiology, development of cells, and pharmaceuticals. Without GFP, these kinds of advancements would be impossible to improve public health and people's understanding of how living organisms function.
Thank you for watching.